In this session, I will be describing about oxidation of hard chain fatty acids. In previous sessions, we have studied about beta oxidation of even chain fatty acids. The objectives of this session are: at the end of the session, the learner shall be able to describe oxidation of hard chain fatty acids, describe disorders in relation to metabolism of hard chain fatty acids. As you know, odd chain fatty acids means odd number carbons are present. So suppose there are many examples are there. One of the example is propionyl coenzyme A. Beta oxidation is same as which is taking place in even chain fatty acids, but the final end product they are different. End products are different. At the end of the finally the oxidation takes place, the end products are different. One of the end product is different. Suppose when you take up the odd chain fatty acid, let us take a presume this is odd chain fatty acid. So odd chain fatty acid oxidation, beta oxidation takes place. There is oxidation, hydration, oxidation, thiolytic cleavage. Okay. Finally, there will be two end products. One is acetyl coenzyme A. It is same as in the even chain fatty acids, but the other end product is propionyl coenzyme A, 3 carbon propionyl coenzyme A. So this acetyl coenzyme A goes to the TCA cycle for complete metabolism. What about propionyl coenzyme A? Now let us discuss about fate of propionyl coenzyme A. This is propionyl coenzyme A. This is what we call propionyl coenzyme A. This one, in the presence of ATP is required, converted to ADP plus PI. One of the B complex vitamin, that is biotin. Okay, there is a fixation of the carbon dioxide is taking place. So by the action of enzyme propionyl coenzyme A carboxylase. So the carbon dioxide fixation is taking place. So biotin is the coenzyme. Leads to formation of methyl melanin coenzyme A. It is called dimethyl melanin coenzyme A. This dimethyl melanin coenzyme A, by the action of methyl melanin coenzyme A racemase, converting one isomeric form to the other isomeric form. Okay, from the dimethyl melanin coenzyme A to the L-methyl melanin coenzyme A. L-methyl melanin coenzyme A. L-methyl melanin coenzyme A. Then the most important reaction in the presence of B12 cyanocobalamin, methyl melanin enzyme is methyl melanin coenzyme A mutase. Then leads to formation of succinyl coenzyme A. You know the fate of the succinyl coenzyme A, that goes to the TCA cycle. TCA cycle. That goes to the TCA cycle. Okay. So when we talk about the gluconeogenesis, we say even chain fatty acids will not contribute to the net synthesis of glucose, whereas odd chain fatty acids, they contribute to the net synthesis of glucose. That is the way this propionyl coenzyme A is there. It is glucogenic. By converting it into the succinyl coenzyme A, so it can enter the TCA cycle. It also can be converted to the oxaloacetate, and from there the glucose can be synthesized. Now the second objective disorders. Okay. So what is the disorder? Methyl melanic acidemia. Methyl melanic acidemia. It may be due to there are many reasons are there. One is deficiency of B12, where it is required at the reaction of mutase, or unable to, even though B12 is supplied, unable to convert okay, into coenzyme. B12 cannot be converted into the coenzyme. Okay. Then the next one is due to lack of enzyme, due to defect in methyl melanate mutase. It may be hereditary, or one is lack of enzyme. The second reason may be reduced affinity for the coenzyme. Methyl melanic acidemia may be acquired or hereditary. Acquired deficiency of B12. Okay. Then hereditary lack of enzyme, absence of enzyme, or defect in the enzyme, or there may be decreased affinity between enzyme and coenzyme. Okay. What are the consequences? Whatever may be the reason, what are the consequences? The increased accumulation of methyl melanic acid in body, in blood and body. Okay. Then ultimately it results into increased excretion in urine. So that is the reason it is called methyl melanic aciduria. Okay. What it causes? Causes severe metabolic acidosis. pH is decreased. Severe metabolic acidosis. Okay. Then damaging the CNS. CNS damage will be the result. Okay. Growth retardation. Okay. Fatal in early years of life. Another disorder is propionic acidemia. Lack of propionyl CoA, propionyl coenzyme A, carboxylase. There may be acquired reason, that is deficiency of. There is acquired reason, so that is deficiency of biotin. Okay. So these are various disorders which are due to the accumulation of methyl malonic acid and also lack of conversion of propionyl coenzyme A. In the series of lectures, we have discussed about oxidation of fatty acids. In this session, we have discussed about oxidation of hot chain fatty acids. Let us sum up. The final end product in even chain fatty acid and hot chain fatty acids, they are different. One is same, that is acetyl coenzyme A. The second one is different in hot chain fatty acids. But the sequence of reactions in even chain fatty acids and hot chain fatty acids, they are same. That is oxidation, hydration, oxidation and thiolytic cleavage. At the end, so there is a production of acetyl coenzyme A, so that can be used in the tricarboxylic acid cycle. The other one is propionyl coenzyme A in the oxidation of hot chain fatty acids. Okay, so what is the fate of this propionyl coenzyme A? Fate of propionyl coenzyme A. Propionyl coenzyme A, in the presence of propionyl coenzyme A carboxylase, CO2 is added, that is carbon dioxide fixation. In the presence of ATP, using ATP, converting it into ADP plus inorganic phosphate. So biotin, a B complex vitamin, is required. Okay, leading to the formation of D-methyl malonyl coenzyme A. D-methyl malonyl coenzyme A, in the presence of racemase, leads to formation of L-methyl malonyl coenzyme A. The L-methyl malonyl coenzyme A, by the action of methyl malonyl coenzyme A mutase, in the presence of another B complex vitamin, that is B12, leading to the formation of succinyl coenzyme A. The succinyl coenzyme A can be used in the TCA cycle. Succinyl coenzyme A can also be used for the synthesis of glucose, as succinyl coenzyme A convert into oxalate state that can be used for the synthesis of glucose. The process, we call it gluconeogenesis. Okay, in that way, we say propionate can be one of the non-carbohydrate substance for the synthesis of glucose. In that way, hot chain fatty acids can give the glucose. Whereas even chain fatty acids, there is no net synthesis of glucose. Okay. Well, disorders. One of the disorder is methylmalonic acidemia. It may be due to deficiency of B12 or even B12 is available, inability to convert to coenzyme. Then the other one is due to defect of methyl malonate mutase. Hereditary, it may be hereditary. Okay, so in that section there is a lack of enzyme or reduced affinity for coenzyme. Okay, what happens if there is accumulation of methyl malonic acid in body? There is increased excretion in urine. We call it methyl malonic aciduria. Okay, the manifestations are severe metabolic acidosis, damage to CNS, growth retardation. It is fatal in early years of life. The second one is propionic acidemia. It is hereditary or acquired. Hereditary is due to lack of propionyl CoA carboxylase. Acquired is due to deficiency of biotin. Okay, manifestations: ketoacidosis, protein intolerance, developmental abnormalities. These are various aspects related to oxidation of hot chain fatty acids. Okay, thank you very much.